Welcome to Cracking Cancer, where we tackle the tough questions about cancer with real patients and world experts. Our mission is to empower the cancer community through education, bring transparency to the oncology system, and drive funding for critical cancer research. Whether you're facing a diagnosis, supporting a loved one, or simply here to learn, we're here to bring clarity, hope, and action to the fight against cancer. Hi, everyone. Welcome to Cracking Cancer. I am your host, Dr. Kyle Concannon. I'm a physician scientist at the University of Colorado, and I am joined today by Samantha Murrell.
Hey everyone, Samantha here. I'm a patient advocate. I have HER2 non-small cell lung cancer. Good to be here. Thanks, Kyle.
Thanks for joining. And today we're talking about what are ADCs, which stands for Antibody Drug Conjugates. This is a relatively new class of therapy where we link antibodies with highly potent chemotherapeutics usually, and we deliver those to people who most commonly have metastatic forms of cancer. And this is topical because Samantha's been on an ADC, the antibody drug conjugate. Samantha, you were on trastuzumab deruxtecan, also known as Enhertu is the drug name. And, you've been on that for a while. You've had some side effects. You've had, I would say, probably a representative experience, but many people are being provided these as options in the clinic. And I think it's kind of a confusing therapy. So we want to get into a little bit of how they work, why we choose them, why they might be better in certain circumstances or unique from other therapies and things like that.
Absolutely. And I have a wonderful list of questions for you, and I'm hoping you can deliver on.
I'm hoping I can too. And we can always, if I say things that are wrong, we edit it out. Yeah, it's the good part of being the controller of the answers that go on air.
So let's start with just the basic understanding of an ADC and how it's different to traditional chemotherapy, because I still call it I'm on a chemo when it's likely quite different to traditional chemo. So I'd love for you to go into the difference.
Yeah, and I think it's probably appropriate to use that terminology when I'm writing about them in grants or other forms of writing. I usually lump them in with chemotherapeutics because the way they kill cancer cells is similar to the way chemotherapy kills cancer cells, although they are uniquely different. So I'm curious, before we get into the nitty gritty, Samantha, when you were first being offered an ADC, what was the description that was provided to you of what these are and how they work?
Honestly, there wasn't much. Just that it was an ADC called Enhertu. I didn't get told really how it works, how it functions. I didn't get given any diagrams or any leaflets or anything. So I was very much in the dark, and someone called Dr. Kyle actually explained it to me on how it would function and attach and do an explosion and kill the cancer. So I very much learned from you how it worked on a deeper level. I did not get given any insight otherwise.
Well, that's I'm happy to hear I helped in some regard, and hopefully it was helpful.
Very much
but
so.
but these are complicated therapies, so I think your experience is probably common, that getting into the molecular detail is probably a lot for a traditional patient visit where you have a lot going on. And our hope is to maybe offload the oncology teams. And if someone is being presented with an antibody drug conjugate, maybe they listen to this podcast, they have some reasonable foundation, so they have a sense of what's going on. Because these things are complicated and it takes a little while to explain it. So generally speaking, we're calling them ADCs, antibody drug conjugates. And what that means is it's an antibody that is pharmacologically conjugated. It's linked to a drug, and the drug is a chemotherapeutic. So really what we're doing is we're using an antibody delivery system to deliver a toxic, what we would call a payload, or the drug, to the cancer cell. So that's what we mean when we say an antibody drug conjugate. It's a form of chemotherapy that is specifically being trafficked to the cancer cell through an antibody. Now, I think a lot of folks, we assume in the medical field that everyone knows what the antibody function is and what the chemotherapy does. And I think this may be a good opportunity to elaborate in a way that we frequently don't have time in clinic. So my hope is this is helpful in that way. But Samantha, maybe I'll punt it back to you. What do you think of in terms of the function of antibodies? Like, what do they do? Why would we be using them here? Do you have a sense from maybe a kind of a more lay perspective?
I'm giving you a blank stare because I really have no idea.
Yeah. So antibodies function, we think of them in the immune system. We get vaccines. Vaccines will promote an immune response, and the immune system makes antibodies. And the antibodies find things that are foreign and they target them for the immune system to destroy them. That's kind of a simplified way to think about it. The way we're using them here predominantly is because they're really, really, really specific. So I can put in something that's unique to the cancer, or at least mostly present on the cancer. And if I have an antibody that binds to that thing, it's going to bind really specifically to that and that thing only. By and large, there are some exceptions, and I'm generalizing a little bit as we always do. But that's why we use antibodies, is because they're really, really specific. And we can have antibodies so specific that if you have a mutation where just 1% or less of that protein is different from normal, the antibody can tell the difference. So the antibodies are really, really, really specific. And that's why we chose this delivery system, because the antibody can have chemotherapy conjugated to it, attached to it. And in theory, since the antibody is so specific, it will go out, it will seek out and find tumor cells and only bind to those. It's not going to bind to anything else in the body, assuming the tumor is the only thing that has the target. So that's why we use antibodies. Predominantly, their function is to deliver the chemotherapy, but the antibody alone also has some activity for individuals who have breast cancer or other different types of cancer. They can attest that they'll sometimes get infusions that's just the antibody. And actually, since you have HER2, the antibody is in breast cancer is usually targeting HER2, and it's just the antibody alone. The antibody goes, it binds to HER2, it disables it, it probably promotes immune system activity, but that is not conjugated to chemotherapies always. So even the antibody alone might be effective. And again, that's because they're so specific. Does that explain the antibody function okay?
Yes, but I have a question that's not on my list related to what you
Oh,
just said.
yeah.
So I understand that there are ADCs for very specific like mutations, but I've also seen where people that don't have mutations are on trials for ADCs. So
Yeah.
how do those work? Or do they work a little bit differently because they're not specifically targeting a very specific thing? So
So
are they any different? I guess my question is.
no, it's a great question, and I love your particular tumor as an example because you have a HER2 mutation. This is a mutation in the protein called HER2, and the protein happens to be at the surface of the cell. The cell is it's a three-dimensional sphere type thing, and you have proteins that are only on the inside of the membrane, so an antibody can't get through the membrane and attack it. And then it has things that are on the cell surface that are receptors. It's similar to, say, the hair on your arm could be targeted by something on the outside, but if I had something that targeted bone, it's not going to get in there unless it penetrated through the skin, unless it gets to the cell membrane. So HER2 happens to be on the cell membrane, which means these antibodies can target it. Now, in lung cancer, we're usually talking about mutations. And a mutation is when an error occurs. In this context, that error makes the cell divide uncontrollably. So HER2 in your tumor has a mutation and it's constantly telling the cell to divide.
Mm-hmm.
However, in breast cancer, HER2 is usually amplified. So it's not mutated. It's the normal HER2 that I have in my cells. The breast tissue that caused the cancer has more of it. So therein lies the difference between HER2 in lung cancer and HER2 in breast cancer. But the antibody is actually the same that we use in breast cancer and lung cancer. So HER2 is a good example because there's not a ton of the HER2 protein throughout the rest of the body. There are some exceptions there, but it has turned out it's actually pretty safe to target it aggressively in both breast cancer and lung cancer. So your antibody drug conjugate that you got, the antibody was just a regular HER2.
Mm-hmm.
And the mutation didn't happen to be significant. It's just that we know your cells expressed that on the cell surface. So the antibody portion was able to combine to it in the same way that HER2 is expressed in a breast cancer that's HER2 positive, meaning we looked and we see it on the cell surface, and the antibody is able to bind to them. So that's the commonality, I would say, between when we use antibody drug conjugates across different tumor types.
Mm-hmm.
And that's just one example of a protein that's commonly, I'll say, targeted. And you raise a good point, because part of the targeting, you ask yourself, are we targeting it because it's causing the HER2 to stop signaling and kill the cell, or does it just happen to be there? So these are, it doesn't really matter as long as it's present.
Mm-hmm.
The antibody drug conjugate will bind and can potentially be effective. Did that answer your question?
Yes, it did.
So maybe we can go a little more into the mechanism then, because we talked about the antibody portion of the antibody drug conjugate, and that is the I'll say the key, so to speak, to go find the cancer. So that's what makes it cancer specific. That's what's different from chemo. Chemo doesn't only go to cancer cells, it goes everywhere. And that's why people get a lot of side effects from chemo. And the theory behind antibody drug conjugates is that if you could take really, really potent chemos and only deliver them to the cancer, then sounds great. The cancer gets the heavy chemo, the rest of your body is largely spared. So you get all the benefits of chemo with none of the toxicities. I can tell you, and you can tell me, that that's not always the case.
Absolutely not.
So we'll get into your experience in a moment, but the reason that may not always be the case is because the antibody is chemically fused to the drug through this linker section, and it's just another chemical that fuses a drug to the antibody, and that chemical has to break down at some point, or else the chemotherapeutic can't do anything. So usually in certain conditions that will break down and it will release the payload. You want that to happen inside the cell, and that's how they're designed. But sometimes that happens outside the cell, which means the highly toxic chemotherapy can go everywhere. And there are trials that failed specifically because that linker was too weak, meaning the chemotherapy just came off and it made people really, really sick. And even a great idea, targeting the perfect thing with a really potent chemotherapy payload, because the engineering of the design wasn't great, or didn't work as well as people had thought. It was toxic, and people probably died from that therapy. I'm thinking of one in particular that I'm not going to name, but just to say, some of these therapies sound really good in principle, but you have to test them in trials, and we do that. And the ones that are probably going to be offered to folks through standard of care have been tested, and we do believe are safe. But this was a good example of a new type of engineering, of chemical engineering, that that sounded really, really good in principle, needed a few tweaks to iron out. And I think once it has been ironed out, it's probably one of the most promising therapies that we have.
Interesting. Yeah, my experience, for me, it was a little different because my first 7 months on the ADC, I was great and it was totally manageable. And then it's like a switch happened on a certain round where it just all went to crap.
Well, and let's back up a little bit. You were prescribed an antibody drug conjugate because you have HER2 mutated lung cancer, and it is approved for that. That was actually the first targeted therapy approved in HER2 driven lung cancer.
Yep.
or at least the first therapy that was specific for HER2 in some way. And uh, what was getting that like? These are infusions.
Mm-hmm.
How often did you get it? What was the like? What should people expect if they're potentially getting an ADC?
Well, I think depending on the cancer and the drug, it can be slightly different, right? But for me, it was every 3 weeks that I would get an infusion. The infusion, I think my first ever ADC infusion, they ran it over an hour and a half to make sure I wasn't going to have an allergic reaction. But then after the first round, I could get it for 30 minutes. I chose to do it over an hour. No scientific justification on why I decided that, but I decided I still wanted to slow roll it a little bit. So I got it over an hour and they gave me a lot of drugs ahead of time. So a couple of nausea meds, a steroid, just to ensure that I didn't have like that initial bad reaction, bad experience from it. And it worked really well, like I said, up to a certain round where my last round that I did was so bad I had an immediate allergic reaction where they had to give me a ton of Benadryl to get my body to calm down. So I not to put people off ADCs by any means, but and I would love to know the science a little bit more on like why some people maybe get bad side effects initially and it just continues indefinitely for them while they're on ADCs, and other people it seems to just get worse, like almost like cumulative, but I was told that it shouldn't be cumulative, so it was kind of confusing to me when I started experiencing these really negative impacts later on.
Yeah, I think there are a lot of types of medicines that can have infusion reactions where your body's immune system is acknowledging that something foreign is in there
Mm-hmm.
and it's revving up. And those are things that the nurses are really trained on. They know exactly what to do. They
Yeah.
know the signs and symptoms, and those are not unique to antibody drug conjugates.
Mm-hmm.
Usually, those are. Well, let me ask you, what was that experience like? Let's lead with that.
Yeah, it was pretty scary. It felt like my blood pressure was just like in overdrive. It like someone had given me 60 steroid tablets, but I hadn't had 60 steroid tablets. I felt really amped up and like my whole body went into like just an amped up high blood pressure, high heart rate mode, and they put on the blood. They immediately, the entire team, because I was like, I don't feel right, and the entire team came. I had like 10 nurses all around me. They put on the blood pressure thing and my heart rate, and my blood pressure was like 160 or something like that. I'd never seen my blood pressure ever be that high. And I know that's not like crazy high, but for me, that was crazy high.
It's going to be someone listening that's like, I wish I had a blood pressure of 150,
Yeah, yeah. So they
which is
immediately,
high. That's a high blood pressure. Yeah,
right, right. And
we would be
so they
working on that.
immediately gave me Benadryl, and within like 15 minutes, I felt like nothing had ever happened. They did stop the infusion while they gave me the Benadryl. And then I started crying because I was like, this drug is extending my life. Like, I need it. I can't have a bad reaction. And they were like, okay, like, we'll take one step at a time. Let's see if the Benadryl kicks in. And then they slow rolled the medicine for the rest of the time, and I tolerated it totally fine.
Yeah, and that's probably a pretty common representation for infusion reactions
Mm-hmm.
is you get your system, you get your body response, you feel amped up, your heart rate goes up. A lot of people will get flushed. I don't know if you
Yep,
got flushed. Yeah.
I did. Yep.
Yeah. So the Benadryl, the antihistamine will kind of calm that down. And then people usually tolerate it just fine.
Yep.
After that infusion, did you have another one?
No, I haven't had one
That was your
since.
most recent. Okay.
Yeah.
Uh,
And ironically, it was number 13. So lucky number 13.
for those who are superstitious out there, or even a little stitious. Um, yeah. So, and a lot of times you can still give the medicine,
Hmm.
but that's different from a true anaphylactic allergic reaction.
Yep.
So there are drugs out there that we have to worry about that with, where if we infuse that, people's mouths will swell, their throats will close. That's not what we're talking about here.
Yep. And I don't
But I
want to
mean,
put
it's still
anyone
unpleasant.
off because, this ADC has done incredible things for me. So I'm very much a proponent of ADCs, but I think it's just good for people to get a perspective of all the things that potentially could happen and be prepared.
Yeah, and infusion reactions, they're not unique to ADCs.
Hmm.
They're present in a lot of what we do in oncology, but they're not pleasant. They are a risk for antibody drug conjugates for sure. So I think anyone who's considering these medicines, consider that might happen, and if it does happen, you're probably going to be just fine. It's just going to be an unpleasant, you know, um, 10 minutes to an hour. That's usually about the timeline. Um, but going back to your situation, what made you start an ADC, and how did it go in the beginning? How did your tumors respond?
So I was on traditional chemotherapy for lung cancer for about 6 to 7 months, and then I started to get some progression in my liver, where I already had spots originally, but they started to grow again. And so they decided that it was time for me to be on a different type of therapy. And this ADC was approved in lung cancer, I believe, only like 2 years ago. But it was there for me, and so I went on it, and I think my first scan was at the 6 week mark, and my tumors just shrunk to oblivion, essentially. It was a pretty incredible response. It was mind blowing for me. And I was like, wow, I am, I guess, actually her too. This was like confirmation that I was, because I had such an incredible response.
And tell me, in the beginning, you had mentioned the side effects were more mild.
Yes.
Yeah, maybe
Yeah.
how are
So
they different from chemo?
so chemo for me, I felt like my whole body was just being attacked. Um, I can't explain it other than I just felt like all my cells were being attacked. With the ADC, I had nausea and fatigue, but I didn't feel like my whole body was on fire. I didn't feel like my skin hurt, or just I just felt like it was very targeted how I was feeling. Now the nausea and fatigue were not fun, and they weren't pleasant. The nausea was quite frankly horrific on my first round, but we found a wonderful drug that I talk about often called Zyprexa, which was a game changer for me and nipped my nausea totally away, basically,
Good.
for the following rounds. But yeah, nausea and fatigue were the big ones on the ADC. And I think they're pretty common for just ADCs in general, right?
Yeah. Yeah, and I think your experience where you had really bad nausea, and were you vomiting?
No, not that round. I ironically vomited a couple times when I was on Zyprexa, but that was on these later rounds that I don't like talking about.
Okay. But to say in the beginning, and some people do,
Yep.
the nausea can be pretty bad.
Yep.
Um, but the care teams usually have some tricks up their sleeve. We don't start most of these regimens that cause nausea. We start them with some anti-nausea meds, but we almost always have other ones. I can't think of an example where we would start with everything we use. So the care team can usually escalate. And then in your case, you escalated. You got Zyprexa, also known
Hmm.
as olanzapine, and that was helpful for you.
Yeah, it was a game changer completely, and I wish more people knew about it.
And tell me a little bit about the fatigue you experienced on ADCs.
It was different to traditional chemo because the fatigue I got on traditional chemo, I felt like my body weighed like 6 times heavier than it actually was. And like walking felt like I was lifting that kind of weight. This fatigue was more like I just wanted to sleep all the time. Um, but I found that if I pushed myself to even just walk down the street, it combated that quite a bit. It's like my body didn't want to move, but when I moved, my body was like, "Yes, I appreciate this," and then I would feel better.
And when did it come on after your infusion? How long did it last for?
So for me, the steroid definitely kept things at bay for quite a few days. It was if I'd have my infusion on a Thursday, the symptoms would start to really amp up on the Monday, and they would last about 3 days, which wasn't honestly that bad. Again, later on down the line, it became like a full week. And it's almost like the steroid became less effective, but initially it was just like 3 days. So it was very much manageable and not that big of a deal.
And so you were mentioning earlier your fatigue was manageable. You had it for a few days.
Mhm.
It started a few days after your infusion, and I think that description is pretty common.
Yep.
Um, and then you had maybe 2 good weeks before your next infusion.
Yep.
Does that sound about right?
Yep.
And how long after starting your trastuzumab deruxtecan did the fatigue get worse? When did the symptoms pick up?
So I want to say probably 6 or 7 months in.
Hmm.
Um, I almost felt like nausea and fatigue almost within 4 hours of my infusion. And then it would go through to the full week. So I was just, nausea wasn't too bad because Iprex was definitely managing that, but the fatigue, not feeling good, the best word to describe it is like really hungover, like the
Hmm.
worst hungover you've ever had. Um, and it lasted a complete week. And so that was really hard for me mentally because then I would only have 2 good weeks and then it would be back again to feeling like crap for a whole week, which mentally was not the best for me.
And did you talk about taking treatment breaks, or what was your care team's perspective on how to manage these symptoms?
Um, we talked about them a lot. We didn't, I'm a type of person that doesn't like taking a ton of medicine. It was a big deal for me to take Zyprexa. So, like, I know some people that have like extreme fatigue get prescribed Ritalin as an example. But we never really had those conversations because I'm more of like, I'm just going to suck it up, which I don't necessarily recommend to people because when you're on a drug like this, or regardless of what drug you're on and you're having side effects, you should be able to take things to manage. But I was very much just sucking it up and like dreading that next infusion. So I don't have any like wonderful advice there other than to speak up and um, ensure that your care team know what you're going through and what you're experiencing and see if they can help.
Yeah, and sometimes you were mentioning the cumulative toxicity. This happens with a lot of different medicines we give, where the body just kind of gets weighed down. I don't have a better description for it. I don't have a
Yeah.
more scientific description for it. Then, it's just getting kicked a little bit, a lot of times over and over again, and you just need a little break.
Yeah.
And taking treatment breaks is almost always okay. And your care team should tell you if you're the circumstance where things are a little rocky, they're not sure they have complete tumor control, and maybe they're hesitant to do that. Uh, but by and large, if your symptoms are so bad that it's really harming your quality of life like that, where you have full weeks that you're just not able to do the things you enjoy, sometimes taking a little treatment break is what your body needs to get back to that baseline
Hmm.
where you were when you started. And you're like, okay, now it's back to 3 days,
Yep.
tolerable, things are okay again.
Yep.
Um, but there's a common belief out there, which is similar to how you felt, that it's hard to alleviate the anxiety of stopping a treatment for any period of time. because I mean, everyone rightfully thinks the tumor is just going to kick up again.
Mm-hmm.
But the truth for a lot of the medicines we give is that they stay in your system for longer than you think. Um, and I'm just speaking in generalities here. It's different for every medicine, but there are a lot of circumstances where if you hold the dose or you wait a week, something like that, it's probably not going to materially change what the tumor is even seeing.
Mm-hmm.
It might a little bit, but not to the point where it's going to dramatically impact care. And there are studies that support this, that treatment breaks don't necessarily worsen outcomes as much as is perceived. And particularly, I mean, if you're having the symptoms that are that bad, there's not a whole lot of other choice. Like you have to be able to live your life.
Yeah, absolutely. You actually brought up something that I wanted to touch on. Because the ADC I was on, it stayed within your system, I believe, for 7 days. Are all ADCs that kind of length of time, or does it completely depend on what ADC you're taking?
Yeah, I don't actually know.
Okay.
So we talk about half-lives usually is how we measure how long a drug will be around. And what that means is I'm going to use arbitrary units here. Let's say you have 100 units of drug that are infused. And let's say your half-life is a week. That means after 1 week, it goes from 100 units to 50 units. The next week, it goes from 50 units to 25 units. Then from 25 to 12 and a half, 12 and a half to 6. So the drug tapers down in that way rather than what we would call linearly. Like it's not like, 50 units go away every week and then 2 weeks it's gone. So the drugs taper down over time. And that's usually how it works. There are rare exceptions to that. Um, so to say, if you delay a little bit, if you wait a week, it's not like the drug is probably going to clear right away.
Mm-hmm.
And sometimes the dosing interval, how long in between doses, will give you a sense. Because if the drug's only in your system for a few hours, we're probably not going to dose it once a month. Um, but these are considerations. Are they different from ADC to ADC? I actually don't know the answer to that because I would imagine it's just to do with degrading the antibody, but I don't know that for sure.
Okay.
There are other considerations, but um, yeah, I'm not sure actually.
All right, let's go back to my question list. Are you
Yeah.
ready?
Oh, hit me.
Would you say ADCs are safer than chemo, or do they just cause different side effects?
Whew, that is a good question. So when we talk about ADCs as a class, we're really talking about two variables or two things to consider. One is the antibody portion, and the other one is the drug portion, antibody drug conjugates. The antibody portions, for you, it was targeting HER2, the protein that's on the surface of your cells in the tumor. But other tumor types use different targets. It'll still be an antibody, but it'll just attack something else. And the side effects that that person may have can relate to what they're targeting. Because, so for you, for example, HER2 is sometimes expressed in the GI tract, sometimes expressed in the heart. And these are things that we monitor for. Now, other targets we're looking at, the antibody will still bind specifically to that thing, but that thing might be present elsewhere.
Mm-hmm.
So the toxicities may be a little different.
Mm.
And then we mentioned the drug part. Now, by and large, these work like chemo. They function by attacking rapidly divided cells. And if the drug breaks off from the antibody and is just in the circulation, then it can work like chemo. And you're going to get fatigue and you're going to get nausea and it can attack white blood cells or, it can impact that. So those side effects can be pretty consistent across ADCs, but there are differences because the drug payload we use is different in every different circumstance. Sometimes we'll use the same ADC, but there are many different types out there,
Mm-hmm.
and they try different drug conjugates, different types of chemo, but most of the time when we're looking at a given disease, we're not trying all of them against that tumor. So, for example, I'll say in HER2, lung cancer, we used trastuzumab deruxtecan because it worked in HER2 breast cancer.
Mm-hmm.
That was it. That was the whole logic, is it worked over there. Oh, the logic as far as I know it. Maybe they
Yeah.
were maybe they had more thoughts when they were designing the trial, but it was available and they used it and it worked, and that's great.
Mm-hmm.
But how those side effects would differ if instead of the deruxtecan portion, that's the chemo, that could have been paired out with something else.
Mm-hmm.
So yeah, a big convoluted way to say um, there are a lot of similarities. The fatigue, the nausea, um, those are pretty common, but they can also be unique depending on what the antibody itself targets, and sometimes even what the payload is, what the drug part is. And that's why it's important when you hear someone's experience with an ADC to not assume that's going to be your experience.
Yeah.
And even if it's someone who has breast cancer, they may have a different experience from someone who has lung cancer.
Mm-hmm.
In lung cancer, we worry a lot about the lung issues sometimes with the ADCs, and in breast cancer they do too. But the fact that there's maybe a tumor in the lung may also change those dynamics.
Right.
So yeah, it's complicated, but your care team should be able to look at the trial that got your ADC approved in your tumor type, and that is often a reasonable starting point for what to expect.
So you touched on ILD a little bit. So I always say it wrong. Interstitial lung disease.
Perfect.
Okay. Um, is that so? There's a high chance of getting it on the drug that I'm on. Is that the case for other ADCs, or is it just specific to the ADC that I happen to be on?
Yeah, so interstitial lung disease can occur when essentially it's a big lung reaction
Mm-hmm.
from the antibody drug conjugate, and it can really affect people's breathing, and sometimes it can be lethal.
Mm-hmm.
I'll say the rate of getting it is not high. We're talking single digit. So it's not a very common reaction, but it's so severe when it does happen that we monitor it. The same is true for heart issues for people who are on some of these too. Sometimes we'll do echocardiograms and just monitor people's hearts, not because we expect it to be problems, but because if it is a problem and you catch it early, the person could be fine.
Mm-hmm.
But if you miss it, they could potentially die,
Mm-hmm.
even if it's very rare. So I'll say usually those types of reactions that are organ specific, like that's a lung thing. Interstitial lung disease only happens in the lung. Those are usually related to the target.
Okay.
Not always, but usually that's related to the target, and is less likely to happen with something that's attacking a different target. So if someone has an antibody drug conjugate that's, you know, treating a bladder cancer, the risk of interstitial lung disease is probably lower.
Okay.
But they may have their own unique risk, and that's something that the pharmacist or your care team should highlight with you. And then you should know, okay, if you're starting to feel shortness of breath, if you're starting to, have a new cough that isn't going away, these are the things to bring up right away. It's not for most people, some of those subtle things, it's fine to let them go on for a week or two and see if they get better on their own. And most of the time for healthy people, they do. But when you're on a new therapy like these, little symptoms can be the early harbingers of something really severe.
Mm-hmm.
So it's important to bring it up ahead of time, because a lot of times we can give steroids, we can do other things, and prevent the really bad thing from happening.
I know we kind of covered this a little bit before, where you said, which was news to me, and you educated me on that healthy cells can sometimes be affected by ADCs when there's like a leakage. I don't know if that was the right term you used, but I can't remember the term you used. But is there anything else that you kind of want to cover there? Like, if there isn't leakage, I'm using this word again, healthy cells never impacted? Like, are people good, or can ADCs sometimes target healthy cells?
Yeah, we have a term for this in the field, and it's called on-target off-tumor effects.
Hmm.
And we use this term when you have something specific like an antibody that is being used against the tumor. So whenever we use antibodies against tumors, and antibody drug conjugates are one example, but they're not the only example. Um, you're you're targeting the tumor because it has on it the thing the antibody binds to. And it's rarely the case that it's truly just the tumor. Sometimes that's true, but usually you have a little bit of that protein in some other tissue somewhere. It's just not enough that it causes problems to give the med. But sometimes it does cause problems. Like the heart with HER2 is actually a reasonable example
Mm-hmm.
where the heart does have HER2 expression sometimes.
Yep.
And well, I say sometimes it has it to the point where it can cause toxicity sometimes. So that's why some people, if they're on HER2 targeting things, antibodies, they'll sometimes get echocardiograms of the heart and make sure the heart's doing okay.
Yep. That was me.
And that's an on target. It's still hitting HER2. It's doing exactly what you designed it to do,
Mm-hmm.
but it's off tumor. So it just happened to be that the amount of your target was up in a healthy tissue. And I'll say, as someone who does research on antibody drug conjugates in the laboratory, we choose these because we think the target is disproportionately high in the tumor. The protein we're going for is higher in the tumor than in other areas of the body. But nothing's perfect. Nothing's 100%. It's not like we screen every human's tissues across every person to make sure you don't have a little of this target in the eye or
Mm-hmm.
wherever. We have databases that show that where people have done similar type of work and they look at the amount of protein that's expressed in all the different tissues, but it's not so granular as that. So it's hard to get 100% accurate with this stuff. And frankly, you have to deal with just the reality of the opportunities you have. We want to make new therapies. We want to get more specific therapies. And if you screened out everything that had 0.1% of that target somewhere else, you wouldn't have anything. So sometimes we have to let good enough be good enough and acknowledge that sometimes we're going to have toxicities with these things as long as they work well against the cancer and as long as the benefits outweigh the risks. But they're not perfect drugs. The target can go up in other tissue types a lot of the time. So people can get diarrhea, they can get other toxicities. And the drug that we bind to that antibody doesn't always only go to the tumor.
Mm-hmm.
We mentioned the case where you're calling it leakage. I haven't heard the term leakage for it, but we can start using it. Uh, but yeah, when the drug comes off the antibody, now it's similar to just having injected that highly toxic chemo.
Mm-hmm.
And the stuff we tend to use is stuff that wouldn't be approved probably otherwise
Interesting.
because it is too toxic. It's way too strong. Although I'll say with the antibody drug conjugates, there is a class of thinking now where maybe a little bit of this leakage occurs, and that's actually where most of your benefit is coming from. It's just the low dose of these really toxic things that's circulating in the bloodstream. Because yeah, well, I don't want to get too much into theory here, but did that answer your question?
It did. It did. And so a lot of people lose their hair on ADCs. In HER2, I was fortunate enough where I didn't, but I know everyone else I know that is or was on it did lose their hair, and I know other people on other ADCs that have lost their hair. Is that the same concept that you were kind of talking about, that it goes after some of those healthy cells and that's why people lose their hair, or is it a different methodology behind it?
I mean, it's probably that. Like when we give HER2 targeting antibodies in breast cancer, and when we give HER2 targeting small molecule inhibitors, the pills,
Mm-hmm.
those don't tend to cause hair loss.
Right.
So it's probably the leakage that you're talking about, where, well, and so let's talk a little bit more about the mechanism of how these things work as well. So what happens is we have several of the chemotherapy payloads conjugated to one antibody, and then we infuse millions of the antibodies. But that's how it is. 5 to 8 or 9. I forget the exact number, the ratio of ADC to payload, but you have several of the drugs conjugated to an antibody, and that differs depending on which one you give. And then since the goal is for the drug to release in the tumor cell, the antibody has to be internalized into the tumor cell.
Mm.
So what that means is when you have a protein on your cell surface, there's no guarantee that protein is going to cycle inside the cell.
Right.
You have to know that. Like that's part of choosing it as a target. And then it gets internalized into the cell, and then any protein that is undergoing recycling in the cell gets degraded. And that's how your cells can kind of keep reusing the same, sources of building blocks. Is it'll break it down into amino acids and it'll reuse that and build other proteins and such. And under that process is when the drug is released. So the receptor or whatever protein on the cell surface has to be present. The antibody has to bind. It has to be internalized into the cell. It has to then be degraded so the drug can be released. And then the tumor cell dies from the drug. That's in theory how these work. But also, it's very possible that when the cell dies, that might release drug too. It just had a bunch of antibodies in it. It's possible that the antibody binds on the cell surface and that doesn't get internalized. So then at some point that's going to break down and the drug may be released. So there's a lot of conceptual ways in which the antibody drug can be bound together pretty well. But if all these other things aren't working kind of perfectly as you envisioned, you might get toxicity from the drug coming off and binding to other things.
Okay.
And the antibody can't go through the cell membrane. It's a big protein. It can't just wither its way through. But those chemotherapeutics probably can. So it's very possible that if they do break off one way or another, and they're just in the bloodstream, you can get your nausea, you can get your vomiting, you can get your fatigue, you can get your hair loss. These are the side effects that I associate with probably the really toxic payload coming off.
Yeah. Okay. That makes sense. I appreciate the explanation.
Yeah. I mean, it's a complicated strategy. And really, it's a really skilled way of delivering chemotherapy, which it's still highly toxic chemos, but we're trying to deliver it in a smarter way. And I think your evidence where you started on a traditional chemotherapy, you progressed, then they gave you this antibody drug conjugate, which is still a type of chemotherapeutic, but it's supposed to be more targeted, and your tumors responded really well. And that's not to say all chemos, we could have tried a different chemo and maybe you would have gotten a similar response, but you got a good response for quite a while. It was the toxicity that made you come off.
Yes, absolutely. Which might be breaking news to people on the podcast that I have come off.
Yeah.
But yeah, I've decided to take a break, and I'm going on a HER2 targeted TKI, which we're doing another podcast on TKIs so people can be better educated on what those are. But a big question for me is, so the ADCs seem to be more common in the metastatic setting. Why is that? Is it because it's so toxic?
I would say compared to many of the other therapies we give, they're not without toxicities, but they're not so bad that we fear them. And there are some therapies that we give that we are worried about. I would say most of the time when a new therapy is coming into clinic, it's coming at a place where you have to go through the appropriate evaluation steps to make sure it's okay to give in a bunch of different contexts. And usually we start in the metastatic setting. Because in a setting where it's not metastatic, if someone has localized disease and they can be cured with radiation or surgery, or if there's really any chance of curing them, you don't want to mess around with that. You don't want to lower someone's chance of cure unless you're really confident it has the potential to work. So you mentioned this antibody drug conjugate has only been approved in lung cancer for a couple of years.
Yep.
We wouldn't start with someone where if we're wrong, they don't get cured. So
Mhm.
that's just how we
That
lay out
makes sense.
trials. It doesn't have, yeah, it's not necessarily about the mechanism or toxicities. I mean, frankly, if it were 3 times as toxic but didn't kill people and it had a higher chance of curing them,
Yeah.
we would go with that.
Yeah.
Um, and there are examples where we do give, I mean, bone marrow transplants, like we give things that are really, really dangerous, need to be done in
Mhm.
the hospital, in the intensive care units. We have really aggressive therapies that have chances of cure. Um, but, it's done in a very systematically interrogated way that we know what we're doing and we know what to expect. Now, I'll say the ADCs have shown a lot of promise in metastatic cancers and cancers that are really hard to treat otherwise.
Mhm.
Because the benefit of ADCs is, in your example, we're targeting HER2 because it's mutated, but it could just be there. It doesn't have to be mutated. There are other targets on your cell surface that may not be driving your cancer, but that doesn't matter with an ADC.
Mhm.
As long as it's there, we can target it. So that's a huge beauty of the antibody drug conjugate class is many of the targets that we use are not biologically relevant. They're not the things that are driving the cancer's growth. They're just there. They get internalized and they're on the tumor and they're not on other things.
Mhm.
And that's what you need. So to your point, we can use those kinds of therapies in different settings too. There's no biologic reason why we can't. It's just since they're fairly new, we tend to start by one, making sure they're safe, and then we make sure they're effective, and then we compare them to another strategy. Um, but I do suspect that for many tumors, antibody drug conjugates are going to go from the metastatic setting, and there are probably already trials doing this. I'm just not aware of them that are going from the metastatic setting into the curative intent strategies,
Mm. Interesting.
um, maybe with surgery or radiation. Yeah, I think that's an inevitability.
Yeah. Interesting. Yeah, I feel like ADC is kind of like a buzzword right now.
Yeah.
It's just like all the people that I know that are on clinical trials are on ADCs. There just
Yeah,
seems to be a lot of clinical trials at ADCs.
there's a lot of potential with them.
Yeah.
Um, but there's also challenges like an ADC will not work if your tumor stops expressing the thing it binds to.
Right.
If your tumor stops expressing the outside portion of HER2 and then it only expresses the inside one that signals, it's not going to bind and it won't work
Yep.
in theory. So there's always that issue. And there's also the challenge where when cells are just tolerating therapy and they're not dividing a lot, they're not very susceptible to chemo.
Mm.
It's true with regular chemo, it's true with antibody drug conjugate chemo. So the cells still have to be active in order to get the benefit and not just the toxicity. So
Interesting.
that's a challenge with ADCs actually.
Mm.
Um, so we still have things to figure out. There's still research to be done. My hope is that in the future we're able to optimize the benefits of ADCs and minimize the toxicities. Like in conventional chemotherapy, we give chemo. Sometimes we'll give it upfront for a few cycles, and then if something's really toxic, we'll stop giving it.
Mhm.
You probably experienced this with the platinum chemotherapy you were on.
Yep.
We know that's toxic, we know it's effective, and we figured out a reasonably good way to give it for a few cycles and then maintain something else
Yep.
that's better tolerated. And we did a lot of trials back in the day to figure out that that's a reasonable way to do it. Um, I'll say by and large the ADCs are less toxic than the platinums, uh, but I think it's early in the field and we haven't settled on the best way we're going to use it for every circumstance yet. We just have more research to do.
Do you think at some point they might replace traditional chemos?
Very possibly. Yeah, very possibly. Uh, it's interesting because it's going to depend a lot on the context. There are some chemo regimens that are tolerated pretty well. There are some chemo regimens that are probably tolerated better than some ADCs.
Mm-hmm.
And then, do we compromise that? It's gonna take a lot of research and work because there are just so many different cancers, and the different cancers have different cell surface targets for them.
Mm-hmm.
So it's a big undertaking to figure out what's the best ADC to use in what circumstance. But I suspect that once that happens and you know the best payload to give, that will probably be better. I mean, I have to believe that will be better than just giving chemo to the whole body.
Right.
Now, the difference sometimes may be the infusion schedules. Some chemos are still pills. So, we have these considerations that are pragmatic and lifestyle considerations. But I think it's hard to believe that having a system that specifically delivers the drug of your choosing to the cancer cell and the cancer cell alone as being worse than just giving chemo to everything, it's hard to believe that that won't, eventually overtake chemo.
Mm-hmm.
I believe it will, but it'll be a while.
Yep. Well, I've certainly learned a lot.
Yeah, and what's a piece of advice you would give to someone who is listening to this podcast and they were just told we're starting you on an antibody drug conjugate? What would you tell them to prepare or how to feel about it?
I would say try and find some kind of online support group that maybe individuals who are already on the drug. You can join and get feedback from those people on what their side effects were like. Although I will disclaimer that, and you've mentioned this before, Kyle, that everyone's different, everyone's going to respond differently. But just getting a general sense of what the side effects might be to prepare yourself mentally and prepare yourself in terms of what medicines might counteract those side effects that I can talk to my care team about. I personally prefer to join groups and talk to other people who have been on the drug rather than read the leaflet that says what the side effects are. But that's just a personal preference for me. I like to learn firsthand from people actually taking it rather than just a pamphlet. While I believe for some people the pamphlet also might be helpful just to be prepared and understand, getting those firsthand experiences are super, super helpful. And I know I've helped out a lot of people who are just starting out on the drug that I was on, and people before me helped me prepare. So it's very much helpful to find other people that are on the same drug.
Yeah, and I'll add to say if you hear about something that might be helpful for the symptoms you have, and bring that to your care team and say, hey, I've heard X, Y, and Z is helpful. What do you think?
Yep.
I bet your care team is going to be pretty receptive. And sometimes, we have the information from the provider perspective from the studies, but it's different reading the grading of a severity, which is a clinical trial term, versus what is the actual experience like. What are the things that really bothered you? We get a sense of that from the trial, but it's not the same as hearing from individuals who have undergone it as part of a support group. So I think, Samantha, what you're recommending, I would recommend for patients too. I think you're going to get more accurate and honest information from those who have trodden that path before
Absolutely.
than the pamphlet.
Yep.
Yeah, and the pamphlet lists everything anyone reported. So yeah, here's 200 potential side effects.
Yeah, which is another reason why I don't like the pamphlet, because for me personally, I almost invent side effects when I believe that I should be experiencing a side effect. And so the pamphlet, because it lists literally everything, I can just invent all these things in my head and psychosomatically they might appear. And I don't want that happening. So that's why I don't like the leaflet.
Yeah, the leaflet, for better or worse, I think of that as they need to describe anything that could go wrong. And you could think of this from almost a litigious perspective, where if someone wasn't adequately warned that they could get a side effect, which is reasonable, they should be warned about any potential side effect. But that just means that the drug companies have to be really, really, really inclusive, which isn't representative.
Right.
Like you tell me, I had nausea and fatigue. Those are my big things. You didn't list 100 side effects, as a normal person who got the drug. So the leaflet's just really hard to interpret.
Absolutely. And another big thing I will say is, if your first couple of rounds are really rough, your care team absolutely can adapt and work with different drugs, whether that be nausea, whether that be fatigue, or what have you, to make your experience better. There's always a way to improve your situation if you're suffering.
Yeah, absolutely. Couldn't agree more. And if you eventually need to come off of this or any other therapy for toxicity reasons, it doesn't mean you can't retrial it later. There's a lot of ways to proceed forward in these paths.
Absolutely. Well,
All
thank
right.
you, Kyle.
Thank you. Thank you for offering all your experience with ADCs. One of the few people with lung cancer to be getting those.
Yep.
So I
Oh,
think
joy.
you're paving the way for many to follow. All right. Well, thanks everyone. And if you enjoyed this, remember to like and subscribe, and you can go to crackingcancer.org for more information. All right. Bye, everyone.
Bye.
Thank you for joining us on this episode of Cracking Cancer. If you found this helpful, please like, subscribe, leave us a review, and visit crackingcancer.org for more information. Every dollar we earn goes directly to furthering the podcast and supporting vital cancer research. Because together, we can crack cancer.